Welcome to Week 2, Chapter 4. In this chapter, we're going to discover the two basic types of cells, prokaryotic cells and eukaryotic cells, what makes them unique, and specifically focus on details of eukaryotic cells. So let's get started. The first few slides are just slides to show you some images of actual cells. So in this slide, we see a prokaryotic cell, that's the rosy pink E. coli bacteria. These next few images are of eukaryotic unicellular organisms, and you can see the diversity of cell shape and cellular structures. So here's a comparison. In A, we have a prokaryotic cell or a bacterial cell, which is in essence composed of a plasma membrane, which is a phospholipid bilayer. In that phospholipid bilayer, we have a cytoplasm. And in the cytoplasm, we have genetic material, DNA, as well as ribosomes. There are also some external features of this prokaryotic cell that help it adhere to surfaces and move. B and C are cellular types, eukaryotic cellular types, which basically means they have a nucleus, they have a specified region for the genetic material, and that genetic material is in a membrane-bound envelope. They also have other membrane-bound organelles, such as endoplasmic reticulum, Golgi apparatus, mitochondria, and as you can see in the chloroplast, as you can see in the plant cell, the green chloroplasts. Here's another view, a zoomed-in view of that prokaryotic cell. By the way, prokarya means basically before nucleus. So this is a cell that does not contain membrane-bound organelles, and because it does not contain membrane-bound organelles, it does not contain a nucleus. Here's a zoomed-in focused view of our plant cell where you can see those chloroplasts very clearly, and you can see the other membrane-bound structures. So here's our membrane-bound nucleus, our endoplasmic reticulum, Golgi apparatus, our chloroplast, and our mitochondria. And this large structure is a storage structure, a membrane-bound central vacuole. In the animal cell, we see a lot of similarities to the plant cell. We have a large membrane-bound nucleus, an endoplasmic reticulum, Golgi apparatus, and mitochondria. Notice we do not have a large central vacuole, nor do we have chloroplast. Basically has been summarized into 4 parts making up the cell theory. And the cell theory states that every living organism is made up of 1 or more cells, that the cell is the basic unit of structure and function of an organism, that all living cells come from pre-existing cells, and that cells contain hereditary material which they will then pass on to their offspring. With that said, one thing that living organisms have to be concerned with, according to their cells, is cell size. As the size of a cell increases, its volume increases at a much faster rate than its surface area. And there comes a point where if the cell increases too much, it'll have too much volume, too much material basically inside of the cell, needing oxygen or needing to get rid of waste, and there's not enough boundary or membrane to do that. That means basically that the cell has exceeded its surface area to volume ratio. So if we look at this slide, imagine we have 3 spherical shaped cells. The one to the left is 2 centimeters, the middle one is 3 centimeters, and the far right one is 6 centimeters in diameter. So basically, as we're going from left to right, we're increasing the diameter of the cell. Along with that increase, naturally comes an increase in surface area and an increase in volume. But because volume increases at a faster rate than surface area, as we increase the diameter of our cell, we are increasing the volume of the cell at a much faster rate than we are the surface area. And we can see this in the surface area to volume ratio calculation. In the cell that is 2 centimeters in diameter, the surface area to volume ratio is 3 to 1. In a cell that is 3 centimeters in diameter, it is 2 to 1. And in a cell that is 6 centimeters in diameter, it is 1 to 1. So basically, as the cell increases in size, the volume increases faster than the surface area. So we end up in the 6 centimeter diameter cell having an equal ratio of surface area to volume. Well, a cell really needs to have a larger surface area to volume to meet all the needs of the organelles that are packed inside of that cell. So surface area to volume ratio limits the size that cells can get. Now cells are small, so we need microscopy, basically microscopes, and able to magnify these structures. The one to the left is a light microscope. The one to the right is an electron microscope. Both of them have their benefits. One of the obvious benefits of the electron microscope is it allows you to see immense detail inside of a structure, specifically inside of a cell. An advantage to a light microscope is it enables us to actually look at cells living and to see natural movements in their natural colorations. So, at first glance, you might think, well, the electron microscope is going to put the light microscope out of business because you can see a lot more detail and a lot smaller internal structures. That's not the case because the light microscope does have advantages as well. If we were to look at the sizes of cells and the constituents or the parts that make up cells, we can see that there is a limit to the light microscope. And the light microscope, although you can see mitochondria and chloroplasts with the light microscope, you can't see the detail of those structures. You'd need the electron microscope with that because the electron microscope lets you see down to their membranous layers. Notice the phospholipid at approximately 1 nanometer in size. And of course, the light microscope also enables us to see objects that we can see without a microscope, but that become even more visible and clear to us with the light microscope.